Welcome everyone, or people of the interwebs, to the first video that is going to be my video blog or vlog of my new favorite science class, AP Bio. AP Bio is very interesting. At Lana High School, we have a really unique science program. In fact, so unique that I'm recording this right now on one of our iPads, because that's what we get when you're in high-level science, is an iPad for you to do all your work on. But it's so much different than any other science class that I've ever done, and I'm learning more each day. I would say I learn more in a day of AP Bio than really I would in a whole quarter of any other regular science class. And yes, it is an AP class, but the difference is that versus being the average textbook-based memorize, then test AP class, we actually do a lot of science experiments, and that's pretty much all we've been doing so far. Right now I'm reaching the end of my first quarter, and I thought I would finally start my work on my blog so that everyone knows what's going on. And we've pretty much only done 3 things this quarter, but there's a lot that goes into those 3 things. The 3 major things that we've been doing are genetics. We've been covering a lot of genetics, and there's a couple different things we've been doing in there. Animal behavioral studies, and then we've also done a major lab on Hardy-Weinberg genetic theory, which is a whole nother mess that I won't get into in this particular video segment, because I'm going to do a text blog all about that later, because there's a lab write-up and lots of data and all kinds of messy things. But instead I'll talk about the fun stuff that we've been doing, which is the animal behavioral studies and the genetic experiments. So for our genetic experiments, what we're doing is we have kind of like a plant colony going on. There's only 7 of us in this class, so it's like really awesome. Basically we go in and there's 8 people in the classroom at any given time, and we just talk and work on our experiments and have these great discussions. I mean, the things we talk about are amazing. Oh, yeah, also we're reading a book called Your Inner Fish, which is all about evolution, and I'll keep updating you guys on that as we go through it. But right now we're chapter 4, chapter 5, and it's an amazing book. I never thought I'd actually like a science-based book this much, even though I love science, and it's written by a paleontologist, and it's all about evolution, and it's just a great book that I recommend you read if you have any interest in evolution whatsoever. But anyway, back to the plant colony. So there's 7 of us, and we split off. There's a group of there's 2 or 3 groups of 2 and 1 group of 1, and we're each raising our own plant colony. Now, my friend and I, Stephen Maloff, who, if you guys have ever watched my earlier stuff from my biology class last year, my final video blog that I made about eugenics was based a lot around research that I pulled from him, because he's great at researching, great at taking notes, great at also capturing everything that we do in class. He takes pictures of frickin' everything when we're doing our stuff, and it's really, really helpful, and I admire him for having that much dedication sometimes. But we're growing a bunch of plants. We've got this cool little setup, and you'll see pictures. I'm going to post pictures and an entire write-up when the lab's finished. The problem is that it takes about a month for these plants to go through their life cycle, so it's going to take us a while, because we're breeding 1 set of plants right now and that just reached their end, and then we'll be breeding another set, combining with another groups to get genetic crosses and whatnot. But what we've been doing is we set them up on this little container, and it's a clear container, and we just put water in it, and then there's a hole in the lid that we put a like a wool almost type substance, and we just put it in and then lay it across, so the water that's in the bottom comes up and soaks through the entire layer, and then we put on top of that a styrofoam little quad, I guess you would say, because it's got 4 squares inside of it, but it's filled with dirt and wicks that connect down to the wool, and then they pull up water. So it's really a really kind of cool aquatic transportation system. But the plants are watered that way. So what we did was we planted the plants and we kept watering them and watching them grow, and went through all the process. And this is just a quick rundown of what we've been doing. But we planted them, watered them, watched them grow. When they got to the flowering stage, we cross-pollinated. So we had 4 different quads, each had a different seed thing in. We cross-pollinated 1 and 2, and then 3 and 4 together. And then we covered them so that they could not cross-pollinate with each other, so that we were getting just the desired genetic mixes. And then we went from there. After the flowers had all died and stuff, and we could see seed pods growing, we removed the covers so that they, because they weren't needed anymore, and we just continued to let them grow, and the seed pods formed fully. And right now what we're doing is we're just waiting for the seed pods to completely dry out so we can bust those open and get the seeds, and then plant a new set of plants that we will then crossbreed with another group, Sarah and... I believe Sarah and Sarah, because we have 2 Sarahs in my AP Bio group. I'll just tell you who the class is. Michael Reese, amazing. He's just got a brilliant mind for science, for math, anything. He writes code for fun. He's a really interesting guy. Stephen Maloff, I told you he's very studious, takes notes like a champion. Myself, I have mad style, and I love bow ties. My friend Danielle Settle, she's really cool, and she's pretty good when it comes to science. Mandy JB, she's brilliant, great bari saxophone player. Then there's Sarah Gallegos, she's a senior, and she's really funny, and she's doing a animal behavioral study on her dogs, because she has like Siberian Huskies. They're really cool. And then Sarah Walter, who grew up on a ranch, so she's got a lot of insight into animal stuff, and it's a really great group just to be in. And then, of course, there's our brilliant teacher, Mr. Ludwig, who is currently heading up our spider containment team, which includes myself, Michael, and Steven, because what we were doing is, when we split off into groups to do animal behavioral studies, it was... we split off into 2 groups of 3 and 1 group of 1. Sarah decided to work on her own because she wanted to do her dogs and stuff. And then it was Sarah, Mandy, and Danielle, and they work with worms. And then Michael, Steven, and I decided we want to do roly-polies and pill bugs and stuff of that nature. And while we were out hunting for all these animals, because we had to go out and collect everything that we needed to get from nature so we could recreate circumstances in which they live and then adapt those for our experiments. But we were out hunting for them, and we were down by the local Presbyterian church because they have a lot of good rocks for roly-polies to hide under, and I overturned a rock, and there was this gigantic web underneath there, and we found a black widow. And we have named this black widow, like we've named almost most things in this class so far, Rufus. Difference is, this is Rufus X, as in Rufus Roman numeral X. We are aware that Rufus is a male name, yes, and that black widows are mainly female. But so we captured this black widow and took it back to our classroom, and we had it in basically what I would call a pill bottle, because it was a container about the same size. And we just had some sticks in there, and it was like that for about a week and a half before we realized, whoa, we should really feed this thing if we don't want to kill it. So I guess one of our animal behavioral things that we figured out was, yes, spiders can survive for about a week or 2 weeks without food, although they do get very irritable and don't move around a lot. So we became the spider containment team. Michael, Steven, and myself, and we've had a lot of fun so far catching grasshoppers and stuff for Rufus's food. But what later on we were having trouble finding roly polies, and so we were kind of just focusing on Rufus for a while. So we built him this really nice container with a lot of just a little bit of dirt and a lot of sticks and really good climbing area and stuff. And left him alone in there for a couple days, and he just makes this amazing web. And like I said, I'll be posting pictures later of all of this. This is just an update because a lot of these experiments are ongoing. So later when they finish, I will, update everything, pictures, videos, all that good stuff. But what we built this really nice container and we left him in there and we've been feeding him grasshoppers. Now, it was super exciting because one day when we decided to do it, we found this amazing or we put a grasshopper in and we captured an amazing video of him killing the grasshopper, or her killing the grasshopper, and moving its web around her, or around the grasshopper, so that like it was wrapping it around it, and it was just so amazing because it's so fast, and it will detach its web in one spot and then reattach it in another like lightning fast. It was amazing. Um, but then after a couple days of just working with Rufus and stuff, Mr. Ludwig brought in a bunch of roly-polies that he found, and I mean like a bunch. We have maybe 20 to 30 of them, and it's awesome because I love roly-polies. They're so cool. If I could, I would grow a gigantic roly-poly and ride it everywhere. I mean, that's like the perfect steed for battle. It's a gigantic armored rolling ball. Who doesn't want one of those? But so we have a roly-poly colony, and we've been doing. We started with one experiment, and it was a light and dark experiment, and we also did a pH level experiment. We put different pH levels in the dirt and left them there for a certain amount of time to see which and like track their movements across a period of I believe 45 minutes, and so we could track which one they preferred better, and same with light and dark. And we've yet to do a second experiment, but we're working on one of those, and we're also finishing up our lab write-ups and data sheets and whatnot for the first roly-poly experiment. But like I said, all of these are ongoing, and it's just been a really good first quarter for science. I mean, Mr. Ludwig said it best when he wrote up a post for his own blog. He said, "We do science," and it really is what we do. We have a lot of fun, and it may seem kind of like we're just messing around, but in all reality, we are doing what people want to do in science, and we're learning all the same stuff that we would learn if we were just taking notes tediously for multiple weeks on end. And we don't lose interest. We're always focused on something science related. May not necessarily be what we're supposed to be focused on for the day, but we're always focused on something science related, and I wish that we could have more time. I mean, day to day, I would spend 3-4 hours in that class because it's such a great atmosphere. I learned so much. I mean, it's just working in that class is fun. That's the only word for it. It's fun. It's enjoyable, and I hope that you guys are excited to follow me through the year. And I think I have to cut it short because, well, not cut it short, cut it long, because it's at 14 minutes and 15 seconds, and I'm pretty sure none of you want to be watching a video longer than that. So I will say adieu until next time, in which you will get a full expose of everything we've been doing with pictures and data and all the goods.